Pilar Alcaide. I am an immunologist at Tufts University in Boston, Massachusetts, and I am here at the virtual Basic Cardiovascular Sciences meeting, and I'm here with Dr. Douglas Mann, who is our keynote speaker for this year's conference. Dr. Mann has made seminal contributions in the field of cardioimmunology and in helping us understand the implications that inflammation has in cardiac repair and heart failure, which is the topic of his keynote lecture. I am very excited that he was selected to be the BCVS 21 keynote speaker, and I'm thrilled that we have this time to exchange, to have a conversation, and he's here to answer a few questions in this important topic.
So Pilar, thanks for asking me to speak today, and I'm tremendously honored to be the keynote speaker at this year's BCVS. The topic I'm going to talk about really started about 30 years ago when we became interested in the concept of inflammation and heart failure. And as you're well aware, there have been a number of fits and starts and bumps along the way. I think the thing that has really advanced the field is the continued focus from basic investigators like yourself who have continued to study the biology. I think when we initially jumped into clinical trials, we probably didn't know as much as we thought we did. And it probably led to some of the negative results that came out. But today, there's so much new knowledge that's been gained that I think our ability to target inflammation in heart failure is rapidly improving. I think there have been some interesting early studies that suggest the benefit, but we're not quite there yet as a mainstream therapy for heart failure, but we'll get there.
So following up on that, I think, as you said, you did this, there was many important findings that for us in the field, we always read the papers from your group and from other groups and trying to merge these disciplines of immunology and cardiology, which I think is critical, right, to understand how to translate basic findings into patients. So what do you think that has been the biggest breakthrough maybe in the past 10 decades that can help us overcome this challenge of translating the findings from experimental models to patients?
I think that the understanding that inflammation probably does more good than harm, right? I think initially we all kind of jumped in and said, oh, inflammation's bad, but then we didn't really realize all the important things it was doing with initiating tissue repair mechanisms. So completely taking inflammation away is a bad thing. And I think our increased sophistication about the connection between innate and adaptive immunity, it has also, I think, enabled us to better understand how to target inflammation. So there have been so many different advances, but I think the growth in innate immunity, the recognition of the crosstalk between innate and adaptive immunity, and then the understanding that the reason that nature retained inflammation was really to help repair tissues. It wasn't there to send people into the hospital and put them on LVADs on a transplant list. It really is designed to repair. And knowing when to interdict and how to interdict, I think are still things that we're working towards. But I think we have a much clearer appreciation today of what the role of inflammation is in the heart than we did 30 years ago.
A related question to that and how we're learning about all these immune mechanisms and how they connect with the heart. How did you become interested in inflammation in the first place? You're a prominent cardiologist, and how did you become interested in studying that in the first place?
Yeah, it was completely by serendipity. I was leaving my mentor's lab, and he didn't want me to take certain projects with me. And so I needed to come up with a new day job. And I remember reading the Milton Packer paper and Beth Levine paper on inflammation and heart failure, and I said, this makes absolutely no sense. How could inflammation have anything to do with heart failure? And my lab was down the hall from an immunologist, Roger Rosen, and he kept on saying, you know, geez, I'd really like to know what TNF does in the heart. I go, Roger, that's entirely descriptive. But then we did a descriptive experiment, and it was fascinating. And then the more I learned and the more I read, I said, geez, there's a story here. And you know, as you know, the biology is so incredibly interesting. And I figured if I was going to fail, I would rather fail reading about things that were interesting than reading about things that I didn't find interesting. So we went all in on inflammation, and we're able to take it to clinical trials. And as you know, the initial trials were negative, if not harmful. And, you know, that caused me to take a step back too and look hard at what, you know, I was doing. But again, I think just always went back to basics, went back to the lab, went back to understanding, because the biology was always right. Just the clinical outcome was not right. But it was really totally serendipity in 2004 when we slipped into this, because I think Peter Liu was the only other person I could talk to, and maybe Peter Libby, who was interested in inflammation and atherosclerosis, but there weren't that many people. So
that's a great lesson. And I think one thing that we continue learning is that heart failure is very complex, and I am an immunologist, so I can see that from a completely view of an outsider. But immunology in itself, I think there's been significant breakthroughs recently as well, that is not as simple as innate and adaptive immunity. Now we know that there are a lot of stromal cells in tissues that actively participate in immune responses. And I think that that could be further leverage in the heart to understand cardiac inflammation, cardiac repair, as you said. You know, that's why our immune system evolved. So in your mind, how do you see? We have a lot of experimental models. We have the benefit of new technologies, right? Single cell sequencing. And there's now massive data on immune cells, stromal cells, name it, right? How do you think is the best approach to use all this massive information that we have and take it to the next level? Because again, then you find the hurdle of that you cardiologists find of how complex heart failure is.
Yeah, I think that single cell data, as you know, is addictive because there's so much information that you get out of it. But I think one thing we need to do is maybe better reconcile single cell data in humans. And I know Corey Levine in our group has been doing that, you know, with some of the mouse models, so that we have a better idea of how the two line up. And at the end of the day, you're going to have to target a cell population or a molecule and put it into a clinical trial and test it. But it's the amount of information coming out is daunting. Today, I'm going to talk a little bit about, we kind of stumbled into immune checkpoints from a different direction. And I think there's opportunity there to learn from how tumor cells suppress inflammation, how we might be able to use that in the heart. I'll touch on that briefly, because you know, nature has evolved all these different ways of handling inflammation, and I still think some of the best ways may be to learn from other experiments in nature. And I think tumor biology, with the ability to turn off the immune system, or at least quiesce it, may be another way to do things. And we have so much knowledge now, thanks to the cancer literature, about how to do this.
Yeah.
But I think there are a lot of ways. And then, of course, getting the right patient population is key.
Yeah, I
and
completely,
that's an even harder question.
I completely agree with that. We need to learn, again, the immune system has evolved over the years, so we need to to understand more what's in nature already. But you touch now on the different heart failure populations, and one question that I have is, what heart failure population do you see benefited more rapidly from either anti-inflammatory or pro-resolution therapies? Because that's the other thing that we're learning. You know, it's not just one type. And what do you think is the heart failure population that will benefit earlier from this?
I think that so there are two answers. I think the acute decompensated heart failure group, the inflammatory signals are quite strong. It's a really vulnerable population. There have been some recent successes in clinical trial design that let you address it. I've been trying to urge companies to get into that space. And I'm not talking about treating the acute heart failure exacerbation the way we've been doing with vasodilators and the different compounds, but rather that patient who comes in the hospital acutely ill who's going home. I mean, the inflammatory signals there are very high. It's a space where we don't really have a lot of drugs. And then the other area is heart failure preserved ejection fraction, whatever entity that is. It's hard to define that, but there's strong inflammatory signals there, at least in the vasculature. So I think that there's lots of opportunity to develop compounds there. You know, unfortunately, the so-called HFrEF space is now really crowded. I mean, there are now 4 drugs that we're starting people on. I don't know that we need a 5th. But as I said, I think the acute decompensated heart failure patient, regardless of EF, and then I think the so-called heart failure preserved EF, if you get the right inflammatory phenotype, there's a lot of vascular problems that those patients get that could be targeted.
Yeah, no, that's one thing that we've all learned, that we need to ask simple questions to get to understand the complexity. But thank you very much for these important insights and for all your contributions to the field that we all look forward to follow up on.
Thanks for the honor of presenting today. Appreciate it.